Great. So thank you very much for coming. So this symposium will focus on how we can effectively integrate neural anatomy and in vivo neuroimaging data, but I will particularly focusing on visual system because I think I'm kind of vision person. So here's the outline of my talk. First, I'm going to start with a little explain what the literature on so-called dorsal and ventral stream in the visual system on primates. Second, I'm going to explain the approach using non-human primate diffusion imaging data to fill the gap across the modalities. So finally, I'm just briefly explain what's the unanswered question from the previous study, then try to explain some multi-modal imaging approach, including high-resolution neural anatomy data. So according to the literature, vision scientists made a great progress in understanding a functional organization of the visual cortex in human or non-human primates. So according to their previous study, there are a series of work proposing a two-stream hypothesis, which distinguishes dorsal stream, which is guiding spatial information or guiding action, or ventral areas, which is interpreting color, form, and objects. So while this type of two streams hypothesis has been widely studied using many methods, so we still do not understand how these distinct systems are communicating each other because they are several centimeters away each other. But in order to support our daily life, so we need integration between such spatial information and categorical information. So visual neuroscientists have attempted to build a wiring diagram or cortical connection in the visual system. So this is a renowned diagram in the Warish and Mobshan paper in 2008. So this is a wiring diagram of the cortical connectivity in the occipital cortex based on a collection of macaque tracer studies. So as you see this picture, so I think the picture is more emphasizes relative separation between dorsal area and ventral area. They actually do not emphasize the connection between them. So because of this kind of idea, I believe that to some extent, maybe visual neuroscience people have recognized that dorsal and ventral stream are distinct systems. However, when we are just carefully checking out classical human neural anatomy, so classical neural anatomists have reported the white matter tract named vertical occipital fasciculus VOF, which is a white matter tract connecting dorsal and ventral occipital cortex. So interestingly, it was already reported in the late 19th century, but it was largely ignored until recent rediscovery in diffusion imaging study. So since it was less underestimated in the literature, so the anatomical detail and interspecies homology of the VOF remain largely unknown. So from 2013, so this is a paper from my former colleague Jason Yetman and Brian Wandel. So they used a diffusion MRI, tractography, and function MRI localizer and find out so there's a pathway VOF located nearby visual waveform area. So since then, we could replicate the VOF finding in the several different data sets. So this is the example, which looks quite similar to what's reported in fiber dissection in the coronal slices, such as VOF connect the dorsal and lateral side of the ventral visual cortex. So nowadays, I think the VOF attracts attention in the field of cognitive neuroscience, and to date, a number of publications report this pathway in relation to the visual function, for example. However, there's kind of big challenges because there's a large gap between the different models proposed by different scientists, because at one hand, the model is emphasizing a separation between dorsal and the ventral stream. At the other hand, so people proposing the large association fiber bundle connecting dorsal and the ventral visual cortex. However, these different models are very difficult to directly compare because they're performed under different spatial scale of the measurement, and also under the different species, and also importantly, since the tracer studies are based on the different injection sites, but MRI studies are much more whole brain, but a coarse spatial scale. So in order to fill the gap between them, so we decided to analyze the macaque diffusion imaging data to improve our understanding of the dorsal-ventral connection in primate visual system. So this approach enables us to understand the organization and test the hypothesis of the macaque visual system as we see in the same data format as we see in the humans. So this is a high-resolution macaque diffusion data from ex vivo brain, then collected by my collaborator, Frank Ye and David Leopold at NIH. So this is a color map depicting principal diffusion direction in each voxel, Then blue color means there's a diffusion going to the superior to inferior axis. So if you are looking at this area, I've highlighted at yellow contour. So there's area of the blue to purple diffusion direction, which is the superior to inferior diffusion. So if we are moving to the coronal slices here, so we can also see a predominant blue to purple color region, suggesting there's a fiber bundle traveling between dorsal and lateral ventral visual cortex. So this color map already indicates that. So there's a similar white matter tract macaque BOF exists. So the nice thing about macaque diffusion approaches is we can just compare it with a wide range of the different invasive anatomical studies performed in a different technique. So this is a comparison I like, but this is a picture in the oldest Kawe Nikki's figure in 1881. So this is based on dissection monkey and horizontal section. So he's reporting that there's a fiber bundle, so vertical occipital bundle, located around here and between superior temporal sulcus and the inferior occipital sulcus. So we can see the location of this blue colored region between exactly the same cell site, replicating the original finding at more than 130 years before. So we can also perform tractography to estimate three-dimensional trajectory of this pathway and also estimate the putative cortical endpoint of this tract with respect to cortical surfaces. So once we have reconstructed this pathway, so we visualize the approximate endpoint of the streamline of the cortical surface like this. Then this visualization enables us to compare the other cortical connectivity studies. For example, Petro and colleagues in 1940s used a method called a strychnine neuronography, which is a strychnine is a glycine antagonist causing a propagation of the electric activity to other areas. So they injected the strychnine to area BTO in the ventral and measure electrical activity induced by strychnine injection. So there are a lot of evidence of the connection between TEO and the dorsal extrastriate cortex, which is qualitatively consistent with what we see in the TEO cortical endpoint, the BOF, and widespread dorsal extrastriate termination. Also, we can perforate macaque brain based on the histology-based atlases, then try to look at which cortical area BOF may terminate. So according to our observation, so BOF in macaque terminate B3, B3A, B4, and MT in the dorsal area, or B3, B4, and TEO in the ventral area. So this observation is consistent with a number of tracer papers, like this is from Leslie Unger-Ryder's paper, reporting the connection, like the bidirectional connection between B3A and B4 ventral using both anterograde and retrograde tracer. So actually, I think the tracer finding and the diffusion-based findings are not really conflicting because both of the data suggest that there are similar pathways connecting the dorsal and the ventral stream. But I believe that when people are just interpreting the tracer data and building wiring diagrams, there seems to be some biases to overlook the connection between dorsal and the ventral area in the primate visual system. So we also analyzed the marmoset diffusion imaging data, very high resolution, then collected by, again, David Ye and Frank Ye and David Leopold. So the marmoset brain looks quite different from the macaque or human because they don't have a lot of number of gyri or sulci, very simple. But still we see the very similar fiber organization, such as optical radiation in green, and BOF-like fiber, which we named the BOF plus, which is a blue color here. Again, it's the same thing in the coronal slices. So tractography-based reconstruction also suggests that, well, there are similar pathways among the human and the macaque marmoset, and the marmoset BOF plus is spanning from anterior to posterior part of the visual cortex, also including parieto-temporal areas. So these connections are very continuous, very predominant. So I believe that the BOF, like a fiber, is one of the predominant features of the primate visual system, not just a little bridges. So finally, I'm going to explain what's the unanswered question today, and then try to explain integration of the possibility with high-resolution neuroanatomy data. So one of the remaining question is, even at very high-resolution diffusion imaging, so it's not always possible to distinguish everything. For example, macaque BOF is located quite nearby inferior longitudinal fasciculus ILF, which is a major bundle connecting occipital and inferior-temporal cortex. So at the level of streamline-based reconstruction, so blue is BOF, red is ILF. So you can see, we can see some complex crossing pattern over here. Then streamlines are trying to intermingle each other. But since this is streamline-based reconstruction, we don't really know how much they are distinct each other or what kind of pattern of the crossing fiber exists in there. So in order to just be more confident about the existence of the fiber, try to understand fiber organization and crossing. So I'm now collaborating with some Professor Karl Zils in Zurich Research Center in Germany to analyze very high-resolution polarized light imaging data collected under bearded monkey. So this is a data using polarized microscopy to measure fiber orientation in the white matter. Then you can see it in the color coordinate here in the sagittal section of the bearded monkey. So this is very high resolution at micrometer scale in in-plane. So as you magnify this area, you can see the BOF fiber coming down, IL fiber from inferior occipital gyrus, they are crossing each other, then exist as a kind of distinct bundle. Also, very high-rich data set with high resolution give us very nice information to understand fiber organization, such as if we are looking at inferior occipital gyrus, we can see multi-layer organization of the ILF with distinct fiber orientation. So if you are more interested in this work, so please come to my poster, TH750 on Thursday. So in conclusion, so a DMRI approach on non-human primates fills a gap between human neuroimaging study and non-invasive anatomical study. So there is a converging evidence that the BOF is one of the prominent features of the primate visual system. Well, and I think that the existing diagram in the visual neuroscience need to be reconsidered. A combination of the DMRI and high-resolution ex vivo neuroanatomy data, including PLI, can help us to further understand the organization of the BOF and neighboring fiber tract. Finally, I would like to thank all collaborators all over the world and also funding agency on funding support from JSPS. And thank you very much.